All right, in this collaborative project between Canopy and Navinci, we performed a multiplex immunofluorescence stain combined with Navinci's PLA on Canopy's CellScape platform. And what we did first is, as a proof of concept, we performed the assay on a human tonsil tissue, where we saw that the PLA signal was mainly localized to germinal centers. And when we look in a bit more detail and utilize the immunofluorescence stains that we combined this with, we saw that the signal is mainly coming from follicular helper T cells and follicular dendritic cells. So the follicular helper cells are PD-1 positive, and the follicular dendritic cells are PD-L1 positive, and they regulate the immune response in these germinal centers. We followed that up with performing the assay on two tumor samples. One is a colorectal cancer sample, and the tumor cells are actually completely negative for PD-L1. But we were still able to see PLA signal coming not from the tumor cells themselves, but from the tumor microenvironment. You can see this cluster of immune cells surrounding the tumor cells. And if we look in a bit more detail, you can see that it's actually tumor-associated macrophages that are interacting with T cells to generate a suppression of the immune response. So they are basically protecting the tumor from the T cells. On the other hand, we have a lung cancer that is PD-L1 positive, about 40%, so pretty high score, which also has a distinct presence of T cells and macrophages as well, but there was no PLA signal present anywhere on the tissue, basically. So what we can garner from this is, on the face of it, this is a PD-L1 positive tumor. The patient would probably be eligible for checkpoint inhibitors therapy, but there's no actual checkpoint activation happening. So this would probably be a bad responder. Whereas in this case, the tumor is PD-L1 negative, but we have checkpoint activation happening in the tumor microenvironment. So this patient might actually have benefited from checkpoint inhibitors therapy. So what we have, what we get here from this combined approach is a very deep insight into the complexity that's defining different tumor entities.